All right, so y'all may be happy to know that yesterday, Tuesday, I went ahead and left my singing on the video. So the video on YouTube, if you want to listen to my terrible singing.
I knew you were going to edit it out.
I didn't.
I knew you weren't going to. That's so much effort.
Well, now I'm going to go put it back in or take it out.
No, you're not.
Y'all just blame Sam. All right, so on Tuesday, we got through all the machinery of the electron transport chain, right? Remember that? We got through all the machinery of the electron transport chain. Today, we're going to finish up the machinery of oxidative phosphorylation. So basically talking about other proteins that are embedded in the inner membrane of the mitochondria, most notably the F1, F0 ATP synthase, okay? And we're also talking about some other important proteins that are embedded in that membrane that allow transport of really critical molecules across the inner membrane. Okay, so as a quick reminder, we know that the chemiosmotic model, or the chemiosmotic theory, is that ATP is synthesized by harnessing chemical energy that that comes from moving protons with their gradient across an impermeable membrane, right? So the idea is that through the process of oxidative phosphorylation, through the process of electron transport, we are moving electrons from our two electron carriers, NADH and FADH2, moving them through a series of electron transport proteins, ultimately giving those electrons to oxygen, reducing it to water. And as we're going to see today, that reduction process, that electron transport process, generates a lot of energy. That energy is trapped by pumping protons from a low concentration inside the matrix out into the inner membrane space where there's a high concentration of protons. Okay, so we harness that energy by moving protons against a chemical gradient and an electric gradient. Okay, so then there is energy trapped in the positioning of protons. And we can harness that energy then by moving them back across the membrane. And when they move back across, they release energy. And that energy is trapped by making ATP from ADP and inorganic phosphate. That's the idea of the chemiosmotic theory. All along the way, from glucose or fatty acids or whatever else the carbon source is, right? We are producing energy by oxidizing carbon. Those electrons are trapped as NADH and FADH2, and we now know that those electrons ultimately are given to oxygen, reducing it to water. That electron transport generates the energy that creates a proton gradient that is then used to drive the synthesis of ATP. Okay? We talked about the importance of the mitochondrial anatomy, right? There's an outer membrane and an inner membrane, effectively creating 4 compartments. Compartment 1 is outside of the matrix, outside of the outer membrane. Okay, that would be the cytoplasm. The second compartment is between the membranes. This is the inner membrane space. The third compartment would actually be the inner membrane itself, because there's a lot of things that are happening inside of that membrane. And then the fourth compartment is inside of the inner membrane, and that's called the matrix. Okay, the outer membrane is highly permeable to most things. The inner membrane is highly impermeable, and the only way to move things across the inner membrane is through carrier proteins, through transport proteins. Okay, a few exceptions are things that can naturally diffuse across membranes, like oxygen. Right, this is an important one. Oxygen can naturally diffuse across membranes because it has nonpolar properties. So it can naturally move with a concentration gradient into the matrix of the mitochondria. All right, so here's another kind of representation of the chemiosmotic theory. We won't go over it again. We talked about how energetically every time we move electrons from one electron carrier to another, it's always energetically downhill, meaning we release energy. Complex 1 is where NADH enters into the mitochondrial electron transport chain. So electrons from NADH are first passed to this flavin mononucleotide. The chemistry of FMN is identical to the chemistry of FAD. Okay, it's able to have a stable radical, which is important because we are transitioning from a 2 electron carrier to 1 electron carriers. all of our iron sulfur clusters in complex 1 are single electron carriers. So the relevance of having this flavin here is that we give both electrons to the FMN, and then it can give one electron at a time to our series of iron sulfur clusters, ultimately passing 2 electrons, one at a time, to coenzyme Q, resulting in a reduced coenzyme Q. The same type of thing happens in complex 2. So complex 2 contains our succinate dehydrogenase, one of our enzymes of the TCA cycle. Okay? Succinate gives electrons to FAD, yielding FADH2, and then it gives electrons one at a time to a couple iron sulfur clusters, which then in turn give electrons one at a time to coenzyme Q. No matter where we start, complex 1 or complex 2, we are giving 2 electrons to coenzyme Q, reducing it to the ubiquinol, their fully reduced form. We then enter into complex 3, where we start with one reduced equivalent of ubiquinol, and in here it undergoes the Q cycle, where one electron goes through a series of B-type hemes given to a different Q site, the Q1 site, and that electron is held onto temporarily. The other electron goes through a Reski iron sulfur cluster, then to a heme type C, and ultimately given to cytochrome C. We repeat that process with another equivalent of our reduced coenzyme Q. One electron goes to the Q1 site, regenerating a reduced coenzyme Q, and the other one goes through the Reski iron sulfur cluster, through the type C heme, and ultimately to cytochrome C. So it's a little bit more complicated process than the single electron transfer in complex 1 and complex 2, but the net result is the same. 2 electrons from coenzyme Q are given to 2 equivalents of cytochrome C. Each cytochrome C carries 1 electron over to complex 4, which is our terminal electron transport protein. Here, 1 electron at a time, we transport electrons through a type A copper site, through a heme A site, and then down to our active site, which consists of a copper B site and a heme A3 site. And oxygen binds between those 2 metals, and that's where it's reduced from oxygen to water. Notably here, we have 4 equivalents of cytochrome C that need to come in one at a time, giving 1 electron at a time to our active site so they can ultimately reduce oxygen all the way down to water. That's a 4 electron reduction process. Okay? We talked about how complex 1 pumps 4 protons. Complex 3 pumps how many protons? You remember? 2. And then complex 4 pumps 4 protons. It might be switched. I might have that backwards. But the overall importance here is that when electrons enter at FADH2, they do not have access to the 4 protons that are pumped through complex 1. Okay? That's important because electrons from NADH result in 10 protons pumped when they go all the way to oxygen. Whereas electrons that originate from FADH2, or we'll see today, coenzyme Q, only result in 6 protons being pumped. Okay? That's important. That actually ends up being very important in the overall ATP yield from these 2 electron carriers. Okay, so now we're going to think about how ATP is synthesized. Right? So generally speaking, we know that it's an endergonic process to link together ADP and inorganic phosphate to make ATP. And anytime we go through chemistry like this, carbonyl chemistry, we always transition through a higher covalent intermediate, right, where we have this oxyanion intermediate. With carbonyl chemistry, we call it our tetrahedral intermediate. Here, it's actually a pentahedral intermediate, right, a 5-coordinate phosphorus. The idea is this is our high energy transition state that then can relax back down to make ATP. So the idea here is that this is not just energetically unfavorable, but also kinetically unfavorable. So the enzyme needs to harness energy to overcome the endergonic process, but it also needs to be able to stabilize this transition state. Okay? So overall, the structure of this enzyme is there's a part that's embedded in the membrane, okay? And then there's another section that's freely floating, well, not freely floating, but that's in the matrix part, right? So it's dissolved in the matrix part of it. They're linked together. The part that's embedded in the membrane is called the F0. The part that's positioned in the matrix is called the F1 section, okay? And the idea here is that protons can move through the membrane through the F0 part in either direction. That's important. They can move in either direction, and they move based on the concentration gradient. So if there's a higher concentration of protons in the inner membrane space side, protons move into the matrix. If there happens to be a higher concentration in the matrix, then protons will move back out. Whichever way is energetically favorable is the direction the protons move. Okay? As protons move into the matrix, this causes a clockwise rotation of the F1 part. It truly, like this is a really cool example of a molecular motor, right? We have energy, which comes from proton pumping, which comes from moving protons from a high concentration to a low concentration, and that energy is harnessed by in moving a wheel, right? So this bottom part, we call it our alpha beta subunit. This actually is like a wheel that rotates around a cog, okay? So the idea then is that moving protons into the matrix causes rotation of these alpha beta subunits, and as they rotate, we're going to see they change structures. And it's that rotation and that adjustment of our active site that allows us to catalyze the conversion of ATP. Okay, so let's go through each part of it. First, the ATP synthase part of it. Okay, I mentioned this is really like a wheel on a cog, and you can kind of see it here, right? So our alpha and beta subunits, right? There's 3 of each, so it's a trimer. It's a heterotrimer. These create the wheel, and this gamma subunit is kind of positioned as the cog down the middle of the wheel. Okay, and what I hope this image represents pretty well is that depending upon how the gamma subunit is interacting with each of the beta subunits, it will cause that beta subunit to be to adopt a different conformation. Okay, and that modestly different conformation really matters. So the alpha subunits are basically just space fillers. They're there to occupy space. They don't play any catalytic role. The beta subunit is where the chemistry happens. And depending upon how the gamma subunit is interacting with it, it can either be present in an O state, an L state, or a T state. Open, loose, or terse, right? So naming convention that's similar to what you learned with hemoglobin, right? The R state and the S state, or R state and T state, right? So relaxed or terse, same idea here. The open state means that the substrates, ADP and inorganic phosphate, can freely diffuse out. Okay, so in this situation here, they are not bound, things actively and quickly diffuse out. The L state, they're locked in place, right? The 2 substrates, ADP and inorganic phosphate, are locked in place, but they can't be converted to ATP. In the T state, now the beta subunit has adopted a conformation that encourages the interconversion of ADP and ATP. Okay, so the idea here is that the gamma subunit in this configuration will create an ATP in this bottom beta subunit, the one in the T state. Okay, there will be an ADP and an inorganic phosphate locked in the L state, and the idea is that ATP has already diffused out of this one, and another equivalent of ADP and phosphate have rebound. Okay, so upon rotation of the gamma subunit, it will rotate 120 degrees, right? So it's going to rotate, so the gamma subunit now is pointed towards this beta subunit, with the one that is blue. And when it rotates, it changes the conformation of the blue one from L to T. The blue one is now able to convert the ADP into ATP. It converts the yellow one from T to O. So the ATP that was made is now able to release, and a new substrate, a new ADP, and a new inorganic phosphate can bind. And it converts the green one, which was O, it converts that to L, so that the substrates that are bound are now locked into place. So each rotation is 1/3 of a full rotation, right? Each time the gamma sodium moves, it moves by 120 degrees. And that causes the production of one ATP. It moves another 120 degrees, the production of a second ATP. Another 120 degrees, a third ATP. So a full rotation of this gamma subunit yields 3 ATPs. Or if it's going in the reverse direction, it will consume 3 ATPs and release 3 ADPs. Yes?
So is that why it makes sense that L would be to the right of T?
Yeah, right. So when the gamma subunit rotates, it always rotates 120 degrees. So then the T becomes O, the L becomes T, and the O becomes L. So as it rotates 120 degrees, the gamma subunit then interacts with the adjacent subunit, the adjacent beta subunit, such that it then will. It already has ADP locked in, so now it moves so it can then convert that ADP to ATP. If it went the opposite direction, things work in reverse, right? If it goes in the opposite direction, this ATP gets converted back to ADP and then eventually gets released. So it's important that it moves in this direction, and that is 100% dependent upon which direction protons are moving across the membrane. If protons are moving from the matrix out into the inner membrane space, if that way is spontaneous, this moves clockwise. Otherwise, it moves counterclockwise, and that really matters. Does that make sense, everybody?
Clockwise.
Well, the punchline is if it moves in one direction, then we are making ATP. Yeah. If it moves in the other direction, we are consuming ATP. That's the really relevant part here. And the direction it's moving depends on the direction that protons are being pumped. Okay? Or moved, as it were. It's not really pumping. All right, so here's kind of a better representation of this. So we start off in the original conformation. We have an ATP present in the T site. We have ADP and phosphate locked in the L site, and then the O site is open. Things can actively diffuse in and out. We get our first 120-degree rotation. The gamma subunit is now pointing to the top right. What was L is now T, so we make ATP. What was T is now O, so the ATP actively diffuses out. What was O is now L, so the substrates are locked into place. We have ATP diffusing out. We have our new substrates, ADP and phosphate, diffusing into the O state, and that resets us for another 120-degree rotation. Now, another 120 degree would make the green one into the T state, the yellow one into the L state, and the blue one into the O state. Okay, yep. That's a good question, and we'll get there. We'll get there, yep. Does this make sense, everybody? Okay. So, I mentioned that this is just kind of a cool little experiment that you can actually do. You can purify this whole complex, and you can anchor it to a slide, right? So you can anchor it to, like, fix it to this this surface through simple, like, nickel histidine chemistry. Okay, and when you do this and you feed it ATP on our C rings, this is the part that will be embedded in the membrane. If you anchor one molecule of biotin and then this molecule avidin, okay, the point of doing this is you can actually visualize this long chain. By doing this, when you feed it ATP, what you end up seeing is this ring rotates in one direction, right? So by feeding it ATP, you actually see the C ring moving around. And that's because as ATP gets hydrolyzed in this direction, because ATP hydrolysis is spontaneous, this enzyme hydrolyzes ATP, it causes the gamma subunit to rotate, and the gamma has to rotate because the alpha and beta subunits are locked into place. And rotation of the gamma subunit causes this C ring to rotate, and that makes this signal spin. And you can actually, oops, I guess I don't have it on there. You can actually visualize that moving 120 degrees at a time. And it's really kind of cool to be able to see it. All right, so how do we pump protons? How do we move protons? And how does that then translate into rotation of this gamma subunit? So the idea here is that this C ring is composed of a number of these C subunits. Okay, each C subunit is 2 alpha helices that span the membrane. And right in the middle is an aspartic acid. And that aspartic acid is important. Okay, and you can think of and then you have this A subunit, which is kind of like, right, so if my water bottle here, right, is the C ring, then this A subunit sits on it like this, right? And the A subunit then links up with the alpha and the beta subunits. So the idea is the A subunit, as we can see here, actually creates a channel, both a channel that's exposed to the inner membrane space and a channel that's exposed to the matrix. So protons can move through this A subunit based on the direction it's spontaneous to move them. So the idea is that when there's a high concentration of protons in the inner membrane space, as there is during active oxidative phosphorylation, then protons will diffuse into the site of the inner membrane space channel. It will protonate this aspartic acid. If there's a higher concentration of protons in the matrix, then the protons would diffuse into the matrix half of the channel and protonate the aspartic acid on that side. Right? So the direction of flow is completely dependent on which side has a higher concentration of protons. Okay? So the way this works is, since there's a higher concentration of protons in the inner membrane space, those protons diffuse through the matrix exposed channel into the aspartic acid, and it binds there. And when it binds, it causes a rotation of this C subunit. And the rotation is by exactly 1 subunit, right? So once this protonation happens, this one right here, let's see if it might be clear if I do it this way, right? So this subunit right here will move over exactly 1 position, making this subunit right here moving over exactly 1 position. And when it moves over that 1 position, note now that in this space, since this subunit moved, there's now a proton that can be released into the matrix. Another proton binds from the inner membrane space, causing rotation, triggering the release of another proton into the matrix. Right? So the idea, and this can go in the opposite direction, right? If there's a higher concentration of protons in the matrix, the protons will bind on the matrix side, causing the rotation in the opposite direction and releasing protons into the inner membrane space. But since there's a higher concentration of protons in the inner membrane space, the protons bind from the inner membrane space. Binding in this position causes the C ring to rotate clockwise, which then releases the proton into the matrix. Okay? The really important thing to recognize here and to remember is that one proton, right, transport of 1 proton causes this ring to move by exactly 1 C subunit. So back to your question, how many protons does it take to get a full rotation? Well, the answer is it's dependent on how many C subunits there are in the C ring. And different organisms have different numbers of C subunits. And the tricky part is that experimentally determining that is actually really hard. So the number of C subunits is absolutely dependent on an organism. But determining that is experimentally tricky. So we can't know 100% for sure how many subunits there are. However, we have a really good idea. Okay? And we'll get there too. All right, so here's kind of a bigger idea, right? So this represents it again. Okay, so our A subunit, we note both the aspartic acids are deprotonated on those C subunits. Okay, so since there's a higher concentration of protons in the inner membrane space, we have a proton binding from the IMS down the channel exposed to the IMS. Okay, now that we have a proton in this position, that will allow the C ring to rotate clockwise. The C ring rotates clockwise. That puts a proton in the channel to be released out to the matrix, resetting it where both of these aspartic acids are deprotonated. When they're both deprotonated, nothing happens. A proton binds from the matrix, or from the inner membrane space. It rotates by 1 unit. The protons are released. It resets. Okay? So if there happens to be 10 C subunits, it would take 10 protons to make 3 ATPs. If there's 8, it would take 8 protons to make 3 ATPs. All right, so overall, this is it. This is the whole process, right? This is taking electrons either from NADH or from FADH2 and moving those electrons all the way to molecular oxygen, resulting in protons being pumped. Okay, so if electrons originate at NADH, it pumps 10 protons, which if there's 10 C subunits in the C ring, that would correspond to 3 ATPs. If the electrons originate from complex 2, then it pumps 6 protons, so it would take more than 1 FADH2 to create 3 ATPs. Oops, why am I repeating this? Okay, so here's another big idea, right? So, and that's how ATPs is synthesized. Does that make sense to everybody? Any quick questions? All right, so what we just covered will allow you to finish the activity from Tuesday. But in the interest of time, because I want to make sure we get through the stuff today, we're going to kind of push forward, all right? So the other really important thing to remember about about what's needed for this whole process is that inside the matrix, right, which is highly impermeable, inside the matrix, we are consuming ADP and producing ATP, right? That's relevant, that's important, because the ATP that we make in the matrix needs to be used in other cellular compartments, right? We don't only use ATP in the matrix. So there needs to be a way of moving that ATP from the matrix out to the cytoplasm, okay? So there's this protein called the ATP-ADP translocase. And the idea is, as it suggests, it moves ATP out as it moves ADP in. Okay, and this is a really common strategy used by these translocases. The one thing I want to really stress about this is with this one and the other translocases we're going to talk about, they are all driven by the electrochemical gradient across the inner membrane. Inside of the membrane, because protons are being pumped out, it's much more negative charge than it is outside, right? Outside, we're pumping protons. There's a lot of positive charge outside, not much positive charge inside. So the inside is much more negative. Note here that ATP has a higher negative charge than ADP. So it is energetically favorable to move ATP out as ADP comes in, because that is going according to our charge gradient. Moving a negative charge out of the cell, moving a positive charge, moving a negative charge across the membrane, and a positive charge the other direction is favorable. Okay? The way this works is ADP binds from the inner membrane space. That triggers an inversion. So what the channel that was exposed to the inner membrane space is now exposed to the matrix. ADP diffuses out, ATP binds, causing the inversion, releasing ATP into the inner membrane space. So it's conceptually simple. The idea is for every 1 ADP that goes in, 1 ATP goes out. And this is absolutely powered by the electrochemical gradient. Okay? Inside of the matrix has a negative charge. Outside of the matrix and the inner membrane space, it has a positive charge. You note here that the molecules that are being transported out all have a more negative charge than the molecules being transported in. Okay? ATP goes out, it has 3 phosphates, more negative charge. ADP goes in, it has 2 phosphates, less negative charge. Malate, remember malate is 2 carboxylic acids. So malate is a -2 charge. Phosphate under physiological pH is a mixture of H2PO4 and HPO4. So its average charge is around -1.5, whereas malate's -2. So phosphate is less negative than malate. Malate's going to go out, phosphate comes in. Same thing, we compare malate and citrate. Citrate has a more negative charge. Citrate has 3 carboxylic acids. malate has 2. So it's favorable to move citrate out, malate in. Okay, so all of these, in all cases, it's really important to be able to get ATP out, ADP in, phosphate in. We'll see that it's important to get malate out. Citrate needs to be able to move in both directions. Okay, all of them are driven by the electrochemical gradient. All right, last big idea before we start thinking about energy. Okay, and that is that most of the NADH and FADH2 that enter into the mitochondrial electron transport chain, most of it comes from the TCA cycle, which happens in the matrix, right? So most of it is generated in the right compartment. However, we've also learned that there is 2 equivalents of NADH that are produced during glycolysis. And we want to be able to get that NADH, we want to be able to get those electrons into the matrix, or at least put them into the mitochondrial electron transport chain so we can make ATP from them as well. There is no transporter across the inner membrane for NADH. So we have to move them in a different chemical way. And there are 2 different ways depending upon which tissue we're in. In muscle tissue, we use something called the glycerol 3-phosphate shuttle. So the idea here is in the cytoplasm, the NADH that we generate during glycolysis or other oxidative pathways can be used to reduce dihydroxyacetone phosphate, right? A molecule we recognize from glycolysis. This is DHAP. We give the electrons to DHAP, reducing it to glycerol 3-phosphate. So the ketone here gets reduced to an alcohol. We convert dihydroxyacetone phosphate into glycerol 3-phosphate. The electrons are now stored in glycerol 3-phosphate. We can regenerate our DHAP using this protein called the mitochondrial glycerol 3-phosphate dehydrogenase, which catalyzes the reverse reaction. But this time, it gives the electrons to an enzyme-linked FAD, which reduces it to FADH2, and then this enzyme then gives those electrons to coenzyme Q. So using this shuttle, we enter into the TCA cycle at coenzyme Q. That's important because we do not then, so we bypass complex 1 and complex 2, which means we bypass 4 proton pumping, right? So we miss out on the 4 protons from complex 1. But this shuttle also prevents the need to pump NADH into the matrix, which energetically is very, very difficult, right? Remember, during active respiration, lots of NADH is made in the matrix. So there's a high concentration of NADH in the matrix and a low concentration in the cytoplasm. So if we had to move that NADH across the membrane, energetically it would be very difficult. This prevents that, okay? But we absolutely lose out on some ATP because we miss out on pumping 4 protons. The other shuttle is called the malate aspartate shuttle, okay? And this one happens in your heart and your liver, okay? So it's tissue dependent. So the idea here is that we take NAD plus in the cytoplasm and we move the NAD, and we have NADH in the cytoplasm and we end up with NADH in the mitochondria. We start with NAD plus in the mitochondria, we end up with NAD plus in the cytoplasm. So there's just simple equilibrium where we shift the position of electrons from the cytoplasm to the mitochondria. It's not as straightforward as that though, right? This is an uphill battle. We do it by moving electrons through other molecules, okay? We're going to start up here. We're starting at NADH. This NADH is produced during glycolysis or some other catabolic pathway, okay? The electrons from NADH are given to malate to make oxaloacetate. We have seen that chemistry before, right? Malate getting reduced to make oxaloacetate is part of the TCA cycle. Malate dehydrogenase, typically we think about as being in the matrix, but there is a cytoplasmic version of it. So we can reduce malate to oxaloacetate, which can then in turn give electrons to aspartate, which can then in turn, let's see, I'm going backwards, sorry. No, I'm right. Okay, so we have oxaloacetate, it gets reduced to malate. Okay, the malate can then move across the inner membrane, right? So there's this protein that can actively move malate in as it moves alpha ketoglutarate out. The malate then gets reduced to oxaloacetate. So I said it wrong the first time when I first said it, right? So the idea here is oxaloacetate gets converted to malate first, okay? The malate then goes into the matrix, which then gets oxidized back to oxaloacetate. So we regenerate our NADH, but now this NADH is present in the matrix. So the left side here is how we have moved NADH from the cytoplasm into the matrix. Okay, we started with oxaloacetate in the cytoplasm. We now have oxaloacetate in the matrix. Our oxaloacetate comes from this transaminase reaction we've talked about before. The idea here is that we can exchange amino groups between two different alpha keto molecules. So the idea here is that aspartic acid gets deaminated and it becomes oxaloacetate. At the same time as alpha ketoglutarate, an alpha keto molecule, gets aminated to make glutamate. So the aspartate becomes oxaloacetate, the alpha ketoglutarate becomes glutamate. This is a transaminase reaction we talked about that uses PLP when we first described our transferases. Okay, so ultimately, aspartate became oxaloacetate, alpha ketoglutarate became glutamate. The glutamate can then can then be pumped across the membrane, so it's now in the matrix. The oxaloacetate gets converted to malate, which gets moved in. Okay, the reverse reaction happens in the matrix. In the matrix, we have glutamate that gets converted to alpha ketoglutarate and oxaloacetate that gets converted to aspartate. The aspartate goes out, which then becomes oxaloacetate, which becomes malate, which goes in, which becomes oxaloacetate, which becomes aspartate, which goes out, which becomes oxaloacetate, which becomes malate, it goes in. Okay? So, you need to memorize this? Nope. You need to be able to look at it and understand it? Yep. Okay? The general idea here is that the only net chemistry that's happening is NADH gets oxidized in the cytoplasm and NAD plus gets reduced in the matrix. This is made possible by carrying electrons in the form of malate, alpha ketoglutarate, aspartate, and glutamate. Okay? Dig it? All right. The beauty of this is we now put NADH in the matrix, so these electrons can now enter at complex 1, which lets us maximize our ATP yield because we pump all 10 protons. Okay? So we absolutely get more energy out if we use the malate aspartate shuttle because we start at complex 1. That now brings us to the energetics, okay? And we have a little over half an hour to talk about this, all right? So the first part of your activity, questions 1 through 3, are things that you have done before, okay? You did it in Gen Chem. We did it a little bit at the beginning of this class. If you took 323 with me, we did it in 323. So the idea here is that you can use these reduction potentials and the number of electrons to figure out the overall energy corresponding to electron transfer between two molecules. Okay, and in the interest of time, let's go ahead and work through the first part. So basically, question A on your activity together. Okay, and we'll talk about some of the relevance here. Right. So remember, as long as you know a reduction potential for an oxidation reaction and a reduction reaction, and you know the number of electrons, you can always calculate the free energy of that electron transfer. Okay? The idea here, when we're thinking about movement of electrons from NADH all the way to oxygen, what we're thinking about is two coupled reactions, right? One of them is the reduction of oxygen to become water. Okay? So oxygen gets reduced to become water. Note here that this reaction, well, half of it at least, is the very, very last thing on the table. Okay? So for each molecule of water that gets produced, we consume half of oxygen. That should make sense. Okay? The potential for this is positive 0.82. And remember that a positive potential is very favorable. Okay? If we're starting with NADH, right? So the oxidation reaction then is going to be NADH becoming NAD plus and releasing 2 electrons. Okay, and then over here we also have plus a proton. Okay, so note here that we have a potential of negative 0.32 for this reaction. But remember, we reverse that reaction, so the potential is actually 0.32 volts for the oxidation of NADH. Okay? When you join these two reactions together, as I show you in question 1A, right? So when we line them up, we see that both of them transfer 2 electrons. If we're dealing with this reaction we see down here, it transfers 2 electrons, and NADH provides 2 electrons. So the idea is we can just add up these reduction potentials. We add up these 2 reduction potentials, and that gives you, what, 1.24-ish? My math right? Okay, 1.14. 1.14 volts. Okay, so the overall potential of this reaction is 1.14 volts. Delta G equals minus NF E naught. The 1.14 volts is our potential. N is the number of electrons, so it's 2. And Faraday's constant is given to you in the activity. It's 96485 joules per mole Kelvin. So you can plug these numbers in and calculate the overall free energy, the overall energy that's released when 2 electrons are removed from NADH and they reduce half an equivalent of a molecular oxygen. Okay, so start there, do that same thing for FADH2, and you're going to see that the energy released is a little bit less because the potential for FAD is 0.22 instead of 0.32. So you're going to see you have a little bit different free energy change, and that's relevant. Okay, and then go ahead and kind of work through the next couple of questions as well.